We want to thank you for one more day you have given in our lives. Lord, we pray that even as we sit here and listen, Lord, we pray that you give us open and willing minds. And Lord, we pray, Lord, for speaker and the moderator. Pray for your wisdom and your insight to guide us, Lord. In Jesus Christ's precious name we pray. Amen.
Good morning, dear BCMCH family. Welcome to this session of Quest. As we look forward to today's speaker, we have a erudite faculty in therapeutic drug monitoring with us. A decade back, when I visited CMZ Vellore for a TDM workshop, I was able to get first-hand information and hands-on little bit of training into what TDM is. And truly, it was cutting edge at that time. And all the more how it has grown over the years, it is baffling. A person who has grown right out of the womb of the birth of TDM in India, the Mecca for healthcare, CMC Vellore. It's a great privilege to have him with us as professor. A year back when he approached us with an ambitious plan to set up a state-of-the-art, one-of-its-kind facility in the country, we were extremely excited, and we embraced him without blinking an eye. And today, all of us stand testimony, and you have to visit this facility in the Medical College block, truly cutting edge and state-of-the-art. So here we are to learn a little bit and get some insights into what TDM is, and as clinicians, as faculty, how we can contribute and how we can build up this facility and gain crucial clinical insights for our patients and for our research and knowledge growth through this amazing facility. Can we put our hands together and welcome the one and only Dr. Sumith K. Mathew, Professor of Pharmacology, Believers Church Medical College and Hospital. Over to you, Sumith.
I'm humbled by the introduction by Jacob sir, and I'm really thankful to the opportunity given for me here in this hospital. And I believe that if this opportunity was not given for me, I would have struggled. Thank you for this opportunity. So I understand that you're all waiting to hear about TDM, but it's not just therapeutic drug monitoring that we are offering in our institution. So what are the areas of our interest? Oh, sorry, it's, can you show the PowerPoint? Yeah. So, the light is not working. I don't. Okay, sure. Okay, thanks. So, in our facility, we are offering some unique services. One is the analytical chemistry service. I will explain that briefly. Then TDM, enzyme phenotyping. I will tell briefly what enzyme phenotyping is. You must be aware about the metabolizing enzymes. For example, TPMT, DPD. These enzymes, we can actually see its activity using genotyping. Okay. Now, another way of seeing its function is actually measuring the enzyme activity. Okay. So in vitro. So we take out the patient blood and we measure the enzyme activity in vitro. And so that is called enzyme phenotyping. It's more sensitive. There can be occasions where we miss some mutations which are important in gene function. If we do genotyping and we may miss them, in such cases, this is more sensitive. Okay? But more specific is obviously the genotyping. So many times we have to combine genotyping and phenotyping to assess the functional status of the enzyme. Then pharmacometrics, another exciting field. Those MBBS students who have graduated but not very keen on the biology, but very much keen on the computer modeling or that things, they find a good niche here. Pharmacometrics, it's all about modeling. Then pharmacokinetics assisted procedures we can do that I will explain briefly later. Model-informed precision dosing, okay, and alternate sampling techniques. So these are the few areas that we are planning to develop in our institution. So first is analytical chemistry laboratory. Why this is so important for the institution? We have a LCMS-MS. It's a mass spec instrument from Waters. And this is a gold standard for measuring any chemical compounds, not proteins, but chemical compounds. So we develop our own assays, and so therefore we can make the assays very cheap. We are not dependent on the availability of kits. We can develop our own assays. It's a big thing. Then so which means if you have identified a molecule of interest and nobody else have done a research on that molecule, we can do a research on that, okay? Follow ICH M10 guidelines for this assay validation, and so there is no issue with questioning of assay quality or anything like that. And this is the only hospital with an LCMS facility in Kerala, okay? And the very few hospitals in country who has an LCMS facility, which may be, there are only 5 or 6 hospitals in a country who have an LCMS facility. So why this? Is it a problem with the money? Yes, of course, the LCMS is costly. It costs around 1 to 1.6 crores for the cheapest LCMS. But is it the money which is a problem? No. The problem is expertise, okay, on how to handle that instrument. That is definitely lacking in our country, okay. This facility we can use for the drug assays, development of drug assays, toxicology, to determine the quantify biochemical parameters, biomarkers, inborn errors of metabolism, but we cannot quantify proteins using this technology. So this is a huge list of drug assays which we are developing right now. What we are going to do is much more than this, okay? This is only for the TDM purpose. And what's the advantage of this instrument, this facility? We can quantify multiple molecules in a single run. So a 3-minute run can quantify maybe hundreds of molecules in 3 minutes. So it's all about how we are developing the assay for that. So it's a high throughput instrument, okay? This whole hospital doesn't need one LCMS, actually speaking, if we develop the assay in that way. Accept different types of matrices. So it's not just the blood, okay? We can quantify CSF drugs in ascitic fluid, maybe even tissues. We can pulverize and take the drug, quantify the drug in that. So it can accept any matrix. It accepts different microsampling methods. You are aware of the dried blood sampling technique, right? So other than that, there are many other devices which are created. We are also creating a device, developing a device for accepting different microsampling methods. Then the more the number of tests is cheaper, okay? And we need expertise in handling this. And over a period of past 5 months, our team, Ruth, Ria, and Denny, they have learned how to operate this instrument. As I mentioned, this is one of the few institutions in the country who have an LCMS facility. So they have learned tremendously in the past few months, and they are an asset for us, okay? So now we'll talk briefly about TDM. Briefly, I cannot cover all these topics in this 40 minutes time. So what I'm doing here is a kind of advertisement, telling you what it typically looks like. Okay, that's all. So what is tedium? Tedium is not an assay. So we often interchange the term tedium and assay. We say vancomycin tedium. It's not vancomycin assay. Okay, assay is a procedure done in the LCMS. That's an assay. But tedium is much more than that. So what is tedium? Tedium is a multidisciplinary clinical specialty aimed at measuring drugs and or metabolites in blood at specific time points to keep the drug concentrations within therapeutic range, and often these are influenced by genomics. So is genomics a part of tedium? Yes. Is monitoring CPK a part of tedium? Yes, provided we relate it with drug concentrations. Okay, so this is called PK/PD. Is measuring antibiotic and determining the MIC of the organism tedium? Yes, provided we link these two. Okay. So, a glance, some few examples of tedium. A few years back, 2014, we did a study in CMC Vellore in few patients. All these patients received 9 million units of cholestyramine sodium and 3 million units TID maintenance dose of colistin. All these patients, 61 patients. And this is the exposure. You see here is the time in hours, and that is concentration of colistin on the y-axis. What we should aim for is shown in the red dotted line. Okay? So what we should aim for is an AUC of something like 50 to 100, or the concentrations of something like 2 to 3 milligram per liter, steady state concentrations. But you see a lot of patients have very high exposure to colistin. And these are all healthy patients, seemingly healthy patients, normal renal function. The clinicians decided to give that dose, okay, according to the guidelines. What is the guideline followed? Based on the GFR, okay? And there are many patients who were subtherapeutic, okay? So this was an eye-opener for the institution because patients were not responding properly to the colistin treatment. Colistin treatment was very common in CMC Vellore. Now they have switched to polymyxin, but even with polymyxin use, they are not able to control the incidence of acute kidney injury. So we have done a study on this. Colistin is a toxic drug. It can cause nephrotoxicity and neurotoxicity. So what we have to do, we have to estimate an AUC over a period of 24 hours. And that's, so what is AUC? So we have given a dose. These are 2 doses given, okay? Concentration is on y-axis and time is on the x-axis. Over a period of time, the drug goes up and comes down, okay? So this area under this concentration time profile curve is called AUC. How many samples we need to take? 8 samples to determine the AUC accurately. But, and especially for drugs which follow 2 compartment models. And this is a tedious task, correct? ICU patients taking 8 samples in one interdose interval, 8 hours, is a tedious task. Not convenient for the patient also. So what to do? So we decided to develop a model. So using Bayesian approach. What is Bayesian? Many of you may be aware of this term. Now new terms are coming like AI, etc. But what is Bayesian? Bayesian is learn from experience. Okay? You flip a coin, you identify, you got the head first time. Flip the coin again, you got head again. Flip the coin 10 times, you got 9 times head, 1 time tail. Okay? So what's the probability of getting a head? It's 90% if you flip the coin next time. So next time, in the 11th time, you will be able to predict what's going to come. Okay? So that's called learning from the experience. And how we use that approach here. So what we are looking at is all the concentration time profiles. This is our bread and butter. Okay? So we give a drug we look at the concentration time profile. For example, this concentration time profile was achieved with a volume of distribution of 10 liter for this patient and drug. Clearance was 20 ml per minute. There are a lot more parameters, kinetic parameters. These are called kinetic parameters. Okay, there's called bioavailability, intercompartment rate constants, a lot of other kinetic parameters. So for one patient, for one drug, a set of kinetic parameters will determine the shape of that concentration time profile. Okay, for example, if the clearance is higher, what will happen? The concentration time profile will shrink. So that patient may be a normal patient. So for different patients, different kinetic parameters, different concentration time profile shapes. Okay? So this is the information that we learn from the past. So we do a study in multiple patients, and it's not only affected by the body mass or genetic profile of a patient, but also by the disease conditions. For example, renal failure. What happens in renal failure? The drug concentrations increase, so the concentration time profile, you will get high Cmax, bulkier AUCs. Okay? What will happen in sepsis? In sepsis, there is capillary leak syndrome. So what is happening? The drug is going from the central compartment to the peripheral compartment, right? So what happens to the amount of drug in the body? And what happens to the concentration of drug in the central compartment? What happens to the concentration of drug in the central compartment? It will come down because it's leaking to the periphery. What happens to the amount of drug in the body? It is high because it's not getting eliminated. It's in the periphery. It's there. So will you change the dose based on seeing the levels of drug? Will you change the dose of drug? Will you increase the dose in sepsis? Of course you need to, because all the important organs of elimination or all the important organs are bathed in blood. Okay? So of course you need to increase the dose of drug. So different factors affect the concentration time profile of an individual. And so what we do is, in Bayesian approach, first step is do a study in a population. Okay? For example, I am doing a study on vancomycin in ICU patients. Okay? So they have a typical set of kinetic parameters. I told you what kinetic parameters are: volume of distribution, clearance, intercompartment rate constant, many sets. Okay, so determine the kinetic parameters and identify something called covariates. So what are covariates? Covariates are factors which influence the kinetic parameter. For example, GFR influence clearance. Albumin can influence volume of distribution. Body weight can influence volume of distribution. So we will quantify or identify covariates for that drug in that group of patients. Okay? And we will represent that with a mathematical equation. For example, clearance is equal to a constant into GFR raised to 0.25. How can we define that equation in a population? By doing a lot of concentration time profiles, checking the influence, all that. You develop a model for the population and use that model that's called Bayesian prior information that you have. And then use that model for a new patient coming to the ICU, and we'll identify that particular patient's kinetic parameters. And that will tell you what dose we have to give for the patient. Okay, so that's called Bayesian approach. Okay. So what are the advantages of using these Bayesian models? We don't need steady state models. We don't need steady state, right? In ICU, we always say, wait for voriconazole, for example, wait for 5 days to reach steady state and then monitor, because after that, we want to reduce the number of dose optimizations we are doing for the patient. So we will wait for steady state normally, but in ICU patients, it's impossible to reach steady state. They are continuously changing. They have sepsis one day is burns, so high cardiac output. Another day there is a lot of fluid loss. So ICU patients are continuously changing. And the advantage of using these models are we don't have to wait for a steady state. And you can sample at any time. You don't have to wait for a trough sample. You can sample at any time, but we just need to know what time it was collected, what time the dose was given. AUC we can see with very few points. It's very cheap because very few samples are required for TDM. Aim for specific target concentration. So I will say, you will decide, okay, for this patient, I want vancomycin AUC 600 milligram hour per liter on day 2. You will decide. Okay, you have to decide and tell me, and then I can tell you what dose to be given to reach that concentration, target concentrations, fast and accurate. Okay. So dose suggestions are, you can give dose suggestions if you use this. So we developed a model for colistin and used that in our patients. We have seen what we do is over a period of 7 days, we have measured the drug concentrations. We taken 3 blood samples and seen the entire exposure over a period of 7 days. And we can even predict what the exposure will be. For example, if the creatinine changes by 2 units, how the exposure will be, or if the body weight changes, or if there is a sepsis, how the exposure will change, we can actually predict. Okay. So we have measured the AUC for the entire past history using just three blood samples, and we looked at the target achievement. So some patients achieved target, some patients did not achieve target. What is the target? We looked at an AUC of more than 50 to 100 milligram hour per liter. It's not one concentration, it's a AUC, okay, over a period of 24 hours. That's the target we aimed at. And we looked at features of clinical failure on day 7. And over the 7 days, the initial 4 days I was interested, and we have decided to go for only 2 days, okay? At least for 2 days, in the initial 7 days, if the patient achieved this target concentration, we will say, okay, this patient has achieved target, okay? Okay, so we looked at the target achievement, yes or no, and the clinical cure. Achieved within 7 days, did not achieve within 7 days. Obviously, this is very few number of patients, 22 patients. So in 22 patients, we could demonstrate that if the patients achieve target, they will have clinical cure within 7 days. You're considerably reducing the time patients are staying in the hospital, in the ICU, okay? So definitely the patient cost will come down. Patients will improve faster. Number of patients who die can come down, okay? And we have looked at the other factors are roughly the similar, okay? Yeah. Then, so we have come up with a protocol integrating TDM into the existing use. So how we do colistin TDM is day 1. So this is TDM. Okay, it's not just measuring a drug concentration. It's not measuring a drug concentration. So you come up with a protocol how to do TDM. Initiation of CMS on day 1. And we have identified a polymorphism which can predispose a patient to nephrotoxicity. That's the first time in the world we have identified that polymorphism. And we will send the blood sample for that, but we don't have to wait for the result for genotyping. Then estimate the AUC, and it has to be aimed for something like 50 to 100 milligram per liter in the initial 5 days. Okay, and then use these Bayesian softwares for dose optimization, and then stop antibiotics by day 4 if the genetic variant is present. Okay, and on day 5 you again do dose optimization to further reduce the exposure between 50 to 60 milligram hour per liter. Okay, and then for how to do follow-up. So that's how we are going to treat the patients in our hospital if you are willing. So the advantage is the number of patients who will have AKI will come down to 0 because of the drug. Okay, now there may be other factors which can cause AKI, but because of drug, so in this previous study, the number of patients who developed AKI was 0 in the patients who achieved target. Okay, even though we may contribute, we're saying, okay, this patient is having that problem, but the actual Drug causing AKI, that proportion is very high. Please understand that. Drug itself is toxic. Then triosulfan TDM, actually, Dr. Chepsi was interested in triosulfan TDM. I just had a reading on that, and this article was published very recently, 2023, in CMC Vellore. And what we had to do is we had to aim for a target again. So what is the target? It's called cumulative AUC of 5,000 milligram hour per liter. What is cumulative AUC? So you give triosulfan over a period of 3 days. So day one dose, day 2 another dose, day 3 another dose. So we estimate the AUC. So combine these AUCs, the cumulative AUC should be 5,000. That's the target. If the cumulative AUC is more than 6,000, that can cause toxicity. If the cumulative AUC is less than 4,000, thalassemia will come back. Okay. So how to do? There is a way to actually decide the dose based on the clearance and AUC for that patient. So the second day onwards, we will do dose optimization. First day, we will decide the dose based on the body surface area and other parameters. But on the second day onwards, we look at the exposure of the first day, we look at the clearance of the drug on the first day and decide the dose. Okay? So finally, by day 3, the AUC should be around 5,000. But the problem is, we have to take 7 blood samples. Even now, this is a problem. These are cancer patients, and getting a line is very difficult, right? So what I'm interested in, and if some of you are interested, this is a SpO2 monitor, you know. So we can just quantify these drugs using infrared. Okay, these drugs have very short half-life, and we are giving only 3 doses in one cycle for that patient. So if we can actually measure these drugs and automate the drug infusion based on the exposure, we don't have to do anything. We just have to give the drug and leave. They will have accurate exposure based on the blood levels. Then this is another test which we have started in CMC Vellore, and it's called indocyanine green liver clearance test. So what normally we do for assessing liver function, we look at some scores, right? But they are all static measurements of liver function, not dynamic measurements of liver function. And therefore, it's difficult to know what is a functional reserve for the liver, okay? So what we do is we give something called, you know, this drug, indocyanine green, is a dye. It is only eliminated by the liver, not eliminated by any other organs. And it has a very short half-life. So we give this drug to the patient, and we will see the levels, okay? And we will find what percentage of drug is retained in the body over a period of 15 minutes. Okay, 15 minutes. So if the retention factor 15, if it is more than 10, we will say that, okay, this patient is at risk. If you do a surgery, hepatectomy, extensive hepatectomy, there is a chance that patient may die. Okay, so these are objective values, and these are very helpful, much better than all the existing scores in predicting liver failure after a surgery or after a liver transplantation. So this facility is also available for collaboration with any other hospitals. As mentioned before, we are developing a device, and hopefully we can use that for getting samples from other institutions if it works. I'm extremely thankful for the institution and the administrators. So I went a few years back, three, 4 years back, begging George sir, can you please give me a job in Believers? And yeah, he said he will think. It took around 2 years, I guess, to finally say yes. I'm really thankful for the institution. And now this is one of the few institutions in India who have an LCMS. And my request, humble request to you is, if you did not find any evidence for TDM for a particular drug, don't worry if you don't have an evidence. It's a good opportunity for you to find an evidence, okay? So please use the facility. And these are the few institutions in the world who have an LCMS facility for a hospital, okay? And I'm wondered to see even most of the analytical chemistry, so institutions offering analytical chemistry degree in India do not have HPLC, forget LCMS. So we can use our facility also for education purpose. And also, I would like to invite you all for the upcoming conference in Malaysia. It's happening in April. It's not too late to register. Please consider registering for this conference. This is all about talk about TDM and clinical toxicology. A lot of international speakers are attending this conference, and our institution is sponsoring this event. Okay, so I saw cross right in the middle. Okay, and I welcome all of you to attend this conference. Can you please switch on the audio? Thank you. Open for questions. Thank you for coming, sir. I mean, I have finished all this so fast so that we'll have more time to interact. So please feel free to ask questions. I'm sure that in 30 minutes' time, I can't tell all the things that is happening in the department. Thank you, Dr. Chips.
I just want all of you to know the success of this depends on you. So unless you ask the questions and until you understand what Sumith has set up, we will end up with nothing. Because this is entirely dependent on us to ensure that he gets to do all that he wants to do. So would you all please ask as many questions as possible? I'd like to hear Sumith struggling with answers. By the way, when Sumith decided to move here, the head of the Department of Clinical Pharmacology in CMC Vellore, where he was working, called me and said, please don't do this to us. So I called Sumith and said, please, CMC is, in my mind, it's still number one. So when they are upset, we won't welcome you. But then later, things changed in our favor. And that's why he's here. He's one of the top people in the world. And we are really privileged to have him with us.
Thank you, Dr. Sumith, for trying to establish this over here. Definitely, we are looking forward to it. I just had two queries. One, don't the labs do it when they get FDA approval for these drugs? These AUC based on the clearances, don't they have already done this prior work? That is one. Second, I understand that this is based on previous set of data that you're creating this approach. So do we need to create our own previous set of data before utilizing this, or using anybody's data is okay to reproduce it? And third is, does it matter if we are using branded drugs versus the generic formulations available? Your colistin data was very intriguing. Did you see that? Was it because of the change in brands, or even if it was the standard brand? I think Vellore only 2-3 brands were used. So did it matter according to that? Yeah.
All questions are good. And uh, the FDA have that information. Okay, that what you said is correct. But FDA cannot reveal it uh, because the companies don't like revealing that information. So why should a company tell, okay, you have to do TDM for my drug, new drug, for dose optimization? How many countries have a TDM facility? So their drugs will not sell if they say that. That's a major point. So they will, nowadays, slowly people are understanding that. So now Pfizer is supporting this TDM event in Malaysia. Right now, companies are not interested in revealing this information. Of course, exposure, the systemic exposure to drug is related to the effect, whether it's adverse effect or actual effect of the drug, which we intend for. So, but what we are trying to do is we develop population models. I have shown you how we develop the population models. Okay, that's exactly what what the companies do for achieving a particular target of concentration in a population. So what they are aiming for is the mean of the population. Okay, but of course there is a standard deviation that they will not reveal. Okay, they don't want FDA to reveal. So that is exactly the reason why we should explore, because for most of the drugs this information is lacking. We need to explore. Please use this facility. This is one of the few facilities in the world where we can actually do this. Actually, you take any new drug, unfortunately not monoclonal antibodies because they are proteins, but all these small molecules. Second question was, I forgot. I'm really bad in remembering things. Oh, yes. So for drugs which are hydrophilic, which many of our drugs are hydrophilic, then we can rely on that model, the existing models which people have created. Okay. So for example, the vancomycin model that we are using is from US, the person who actually created Bestos. And we have validated that model in CMC Vellore and it's working perfectly. Where we should be worried is mainly the population. We have to see that it's not pediatric population, which population it is, where the model was developed. And also drugs which are metabolized, of course, we have to validate those models in our population because genetics may play a role. So of course, yes. Yes, sir.
Our population is a South Asian population.
Yes, sir.
So the genomics are
Different.
drugs developed in the Western population. Hopefully they were experimented in Indian South Asian population also.
Yes.
So the metabolism of the drugs and other things are varied and the
Very true.
drug level also varies. What is your
Very true, sir.
Another point is many of the drugs we are manufacturing from high quality pharmaceuticals to low quality pharmaceuticals to generics. So people ask me that we can rely on that particular drug and what is your opinion regarding this?
Yes. That was the third question also asked. So this graph is not coincidence. This is our patient graph actually, which we run in our hospital. We did not reveal this information. We are just trying all these models in our population without your knowledge, okay? So we are getting patient samples from ID department. So we inform the ID, all the concentrations. And this is a model developed in US, again, another model developed in US for voriconazole, okay? And see how exactly it is fitting. These are the two blood samples that we have received. Voriconazole is a drug metabolized by the liver. So of course, we have to validate the models, especially the ones which are metabolized. That models we have to validate in our population before we use it for regular routine practice. Definitely, yes. And yes, sir. So this year, 2023, in Jan, we have started seeing many patients who were given posaconazole And posaconazole use was very common in CMC. It's very common in CMC Velo. So ID use it commonly, hematology use it commonly. And suddenly we have seen a lot of patients are getting under exposure from January 2023. And that we continue to see for 3 months. So then ID got a little upset. They consulted us. They checked our assays. Our assays were okay. But then patients are also not improving. So then we looked at the brands. So we were in the TDM lab, we always collect the brand information also. Okay, so especially for drugs like this, which have a bioavailability issue. So we have identified that only 2 brands out of 7 were achieving target concentrations. So many of these Indian brands are not achieving target exposure. So this we found because we are doing TDM in our hospital. So we found that information. So we could actually stop those brands from prescribing. Yes, it can happen. But to majority of, these are the problems only for drugs like highly lipophilic drugs, which are difficult in getting absorbed. Bioavailability is generally less for those drugs. So those drugs especially are in danger.
Genomics and the selection of the drugs in the future.
Yes, sir.
This may club together, because initially check the genomics of the patient. Then selection of the drug may be AI assisted.
Of course, yes.
Recommendation may be from here.
Definitely, yes.
Giving a highlight.
So even that information helps. So if you know the genomic information, and we know that genomics is affecting the clearance of the drug, then actually the first dose, loading dose to be given, we can decide using the. So we have to, of course, send the genomic to blood test to the lab, and also we can use these programs to decide. Okay, these mutations are there. So what is the first dose I have to give? We can use these programs to decide that, without any blood concentration data. Yes, ma'am.
So I was just thinking a little not related to infections and sepsis and things like that. I was just thinking about what about the role of TDM in NCDs. So what I had in mind was when you were talking, I realized about the rule of halves in hypertension, wherein only half of the people who are treated for hypertension, they have their hypertension, their BP control. And I also realized that when you do an ambulatory BP monitoring, we find that their BP levels shoot up at unexpected times. So I was just wondering if we could do this to see if the people we put them on antihypertensive, do they attain the therapeutic dose? And is it, or do they or don't they? And is it one of the reasons why their BP is uncontrolled? Would that be something good to pursue?
Of course, we can actually pursue that as a research, not a problem. We have to identify what is the therapeutic range for these antihypertensives.
Yes, sir.
So
as
if
you were
it is
saying,
established, we can actually see whether these patients are achieving these concentrations. Of course, the formulations also can be a problem.
exactly. So that was why I was thinking. So you said 2 out of 7 drugs used for some. Posaconazole. So what about? Let's look at some of the brands that are usually given at the government facility and at the private facility, and why don't we compare which drugs attain a proper blood level to reduce their blood pressure? Could that? So I was just thinking.
Yeah, so pharmaceutical equivalence is one factor which we look at. So if you give me 7 different brands, tablets, and I can actually quantify the amount of drug in these tablets. That's one way actually looking at it. But when we did that posaconazole, all the 7 brands had adequate levels of concentrations of drug, amounts of drug there. But it's not just that. How they prepare the tablet also matters in bioavailability. Okay, so yes, even we can actually look at patient blood levels and of course, that can be done as a study, no problem. Yes, thanks. Yeah, that's a good option for a study. Yes. Good morning, ma'am. Yes.
From the practical aspect, can we start sending samples now for our patients?
So for antifungals, we have started the service, but we have not officially
For the antibiotics.
revealed that for antifungals.
Okay.
Voriconazole, itraconazole, isavuconazole,
so only for antifungals as of now.
posaconazole, yes,
So what I want to know is,
and amiodarone.
as a plan for our institution, is it that you're going to routinely take like what you're doing now and tell us what would be the ideal dose, or on a
Okay,
patient basis, we have to send every time when a patient is admitted?
Yeah. So how the logistics are going to be?
Yes.
Okay. So we have a lot of families working in our institution, so we are going to use that potential. Of course, it's difficult for me and for you to see all the TDM samples, when to do it, when should be the follow-up. So we are developing a system where the department will take care of TDM for that department. So in medicine, you have PharmDs, I guess,
Yes.
and we will be. So if you think that okay, this patient needs TDM for this drug, you have to inform your PharmD person, and that person will inform us. Okay, then they will enter the details in the database for the precision medicine unit, and we will, once we give a report to you, we will tell when the follow-up should be, so that the PharmD person will inform you. So the initiation of TDM surveys will happen from the department, and same is the case for OPD patients.
So
as
they
well.
will guide us when to send the next sample.
Yes,
and all
yes,
those
and we
things.
will communicate to them.
Okay.
Even dose optimization, so I mean, which dose we will suggest only. We will only suggest. So all these suggestions we will actually put up in the.
Because we thought if we can
Database.
start from the ICU areas where the patients
Yes.
are really critical,
Yes, ma'am.
that's where really it matters the dose levels
Yes,
for us.
yes. Right now we are thinking of only starting the antifungal service because we are interested in actually finishing off all the assays development. So unless we do that now, once we start the service, then it will be difficult for us to do the development, assay development. So right now focus is only on antifungals, and amiodarone is also developed along with it because it's chemically similar to antifungals.
Thank you, sir.
Thank you, ma'am. I hope we are following a rule. We decided to develop drugs based on their log D characteristics. It's a chemistry. So we are just following it, and we are happy to see that if we follow that pattern of doing assay development, we can actually do the developments faster. So thankfully, we are experimenting, and all these drugs, there is a success. So I hope by May we should finish assay development for most of the drugs. Anti-cancer drugs, we have an issue with the fume cupboard. So anti-cancer drugs we have not started yet. So I think that will be sorted out soon. Yeah, that's the timeline.
Nearly 10 to 20% of the consultations for the nephrology department is for antibiotic dosing for whenever the creatinine is high, and most of the time we write a dose which we are also not very comfortable because the creatinine and the actual kidney function do not always correlate. And can we expect in near future where we do not be called and you have all the equations and you can predict it more precisely than us? Is it possible?
We don't want to replace any existing way of function. So there is no consultation for us. So I guess the same department can be called on for. So these are, it's not shown. Can you show the screen once again?
I mean, some predicting equations that can better predict the dosing than just a creatinine-based dosing that we just look at the reference and write it in the consultation sheet.
See, that's the thing. So it's not connecting, is it? Yeah, there is a list of antibiotics which we are interested in. And not all antibiotics we can help with. So these are the antibiotics we are focusing on right now, antifungals, anti-infectives. But for dose adjustment, it's not just anti-infectives. I guess even for immunosuppressants, for many drugs, AKI can be a problem. And yes, actually we can give you dose suggestions, but I guess a TDM should be a multidisciplinary approach. It has to be that way. For example, in ICU, I would love if an ID physician actually see the patient if there is a infection. So it'll be easier for us also if you develop, if there is an existing system, I would like to actually use that facility, that system to report to reach the patient. So if there is a system, if nephrology is actually doing that, then if you continue doing that, you send the samples, maybe the nephrology takes the initiative in sending the samples to us, we will report to you. So I don't want to affect the existing systems of function.
Just a moment. Thank you very much. It's just a couple of questions regarding the beginning from the ICU where you would like to concentrate. Okay, do you ever envisage, you said a higher dosage of antibiotics are definitely essential in sepsis because of the low albumin levels. But these patients usually get albumin infusions in between, some quite frequently and some maybe once in a week or once in 3 or 4 days. And some every alternate day, some daily. Do you envisage a change in the antibiotic dosage on the day or the day following albumin infusion, or would you just be content with carrying on with the same dose? Number one. Number two, the ICU patients contribute to just a minor fraction of the patients we see. Most of the drug interactions and everything take place in the OPD basis where we prescribe a lot of drugs without having the facility to monitor. Would you envisage such a, for people who are generally on anti-tubercular drugs or some antibiotics prone to cause side effects, maybe fluoroquinolones, or be hesitant to use gentamicin, would you be able to predict the possibility, or anti-tubercular drugs, the possibility of these people going or heading on for problems because of the drugs?
Yes, sir. Okay, of course. So I have listed levofloxacin, amikacin, etc. in the anti-TB drugs. So rifampicin exposure is one interesting study that we have done in CMC Vellore. In children, we have done a study. 86% of children achieved subtherapeutic levels of rifampicin. And one of the reasons why maybe the TDM is, the TB is so prevalent in our country may be subtherapeutic exposures. And Dr. Blessed, he developed a model for rifampicin, and he identified a dose which is much higher than the existing dose of rifampicin to be given for children for the population. Now, I'm not talking about TDM dose individualization. For the population, current dosing recommendation is something like, I guess, 10 or 15 milligram per kg, I guess. And he is suggesting a dose of 35 milligram per kg. And at that time, there were not many studies on that. It happened in 2016. But now, a lot of studies have come up saying that even for adults, we need much higher doses of rifampicin. So yes, TDM for TB is an important aspect, especially for rifampicin. And even for OP patients, we will cater TDM service. I hope I answered that. Yes. And albumin infusions. What we look at is if the drug is not highly protein bound. Highly protein bound means more than 90% protein bound. If the drug is not highly protein bound, we will actually see the total concentration of drug. So still we actually go by the total concentration of drug to say that the exposure is adequate or not. So the advantage of having a TDM facility is irrespective of the albumin infusion times or irrespective of the dialysis time, we can ensure adequate exposure to the drug throughout the treatment period. So you can decide when to do dialysis based on that. You can decide when to do an albumin infusion or decide when to give a next dose of drug based on that, irrespective of the time of albumin infusions or dialysis.
Just in continuation to the answer for Dr. Arun's and Dr. Jensi's question. So while I was in Canada, in Toronto, so for all the patients who are taking vancomycin and amikacin or the aminoglycosides, you have to do a therapeutic drug level, and based on that only you can give the next dose. So you have to maintain the vancomycin dose maybe in between 10 to 15, or if you want a higher target, maybe up to 20, but not more than 20. So based on that, the next dose. So if you're giving 1 gram every day, and if your levels are say more than 20, the next dose should be reduced to 500 milligram. And then before the next dose, you can do the therapeutic drug monitoring and again adjust the dose. So for certain medications, you want to do, or you definitely need to do the therapeutic drug assess and then adjust your dose. When I came back, it was very difficult for me to dose the vancomycin and amikacin in a random way. As Arun was saying, we cannot do that on a random way. We have to use the therapeutic drug monitoring facility and adjust the doses to avoid the side effects. Definitely.
Yeah, so once in 3-day administration of vancomycin may not be the way once we start the TDM survey. So we look at, okay, if the level AUC has gone to 700, now we have to wait. I will suggest you the day, next when to do TDM. So we make sure that the levels have come down enough to give the next dose. So that's the way is not more following the rules of once in 3-day dosing or once in 4-day dosing. Yes. Thank you.
I have a little suggestion for all of you clinicians, because this IIM Ahmedabad encourages a lot of studies looking at cost effectiveness. So if all this gets ready in 3-4 months' time, this is the time to look at your drugs that you're using now. Use that as your pilot study and see how much you're spending. And then once this effective, once he puts all this out, you will obviously use the correct doses. And can you see, you'll probably find it's much more cost effective. So we are reaching a stage where cancer therapy people talked about targeted therapy. Now we are going to make it hopefully available to the whole country, that we can use appropriate drugs in the right doses. So thank you very much, Sumith. Thank you very much. And Ruth and the team, your team is here? Congratulations for all the work done so far. Continue to do very well. Thank you. I've got 2 pieces of good news to tell you. 3 of our stalwarts had got very lucrative jobs abroad. One of them is Gio, who had one of his friends offered him an excellent job in Mauritius. So I was sad. We were all sad. But suddenly he came and told me, said, I don't want to go. I want to be part of all the excitement that's happening here. Thank you for staying back. Dr. Jacob Jesuran and his wife, Lydia, had excellent jobs available in Malaysia. Yesterday evening, 5 o'clock, he made the decision that he's not going. Both of them are staying back.
Thank you, sir. That was a bolt out of the blue. Thank you. Having postgraduates at any institution is a feather in the cap. And in continuation of postgraduate degrees being added, postgraduate courses being added to our institution, this year we have a set of new postgraduates joining us, and we are excited to welcome them. As I call upon the names, can you all please come forward? Ashley Ipin and Ameya in MD Anesthesiology, Devika and Akila in MD Emergency Medicine, Gopika and Jacob Zachariah in MD General Medicine, Malavika and Irene Elizabeth Varghese in MS General Surgery, Athena Thomas, MD Microbiology, Neetu Venugopal and Agna in MS Obs and Gyn, Sherwin Anthony, Sebi and Sarah Maria Thomas in MS Ophthalmology, Rinku T. George and Karthik in MS Orthopedics, Anu K. Sajan and Amrita in MD Pathology, Ashish K. Samuel and Vivek Ajit in MD Radio Diagnosis. It's a great privilege to have you all with us, and may I request our director to say a few words.
No, he rattled off a lot of names. We'd like to know who is who. So radiology can start.
I am Dr. Ashish K. Samuel. I am coming from Kollam. I did my UG from Amala Institute of Medical Science, Thrissur.
I am Dr. Vivek Jit. I am coming from Avilikara and did my UG from Trivandrum Medical College.
I am Dr. Ashley. I am coming from Kottayam. I did my UG in MOC Medical College, Kolancheri.
I am Dr. Devika, coming from Trivandrum. I did my UG in Sri Mukambika Institute, Kanyakumari. I am from the Department of Emergency Medicine.
I am Dr. Akhilash Shaju and I am from Thrissur, and I did my MBBS from Amala Institute of Medical Sciences, Thrissur, and here MD Emergency Medicine.
Good morning. I'm Dr. Jacob George. I did my MBBS from Belgaum, JNMC, and I'm from MD General Medicine Department.
I'm Dr. Gopika Gokul. I come from Kannur. I did my UG in Baroda Medical College, and I'm first year PG resident in General Medicine.
I'm Dr. Adina. I'm from Changanasheri. I completed my MBBS from Pushpir Institute of Medical Science. I'm here in MT Microbiology.
I'm Malavika, PG of General Surgery Department. I'm from Thrissur. Studied my UG in Mahatma Gandhi Medical College, Pondicherry.
Myself, Dr. Agna John from Thrissur, completed my MBBS from Sri Gokulam Medical College, Trivandrum, OBG department.
Good morning, I'm Dr. Neetu Venugopal. I completed my UG from Amala Institute of Medical Sciences, Thrissur, and I've joined here as first year PG resident in OBG.
I'm Dr. Amrita K. Wilson, coming from Ernakulam. I did my UG in Dr. SMCSI Medical College, Karakona, and here in Pathology Department.
Good morning, I am Dr. Anu K. Sajan. I come from Trivandrum. I did my UG in Kannur Medical College and I am in Pathology Department.
So I can see some bright-eyed people like Remy and all that sitting there. It's great to have postgraduates because the level of discussion, learning, training changes completely. So as I told them during their induction, when new children join us, it's like when a new child is born at home. So that's the excitement we all feel. Welcome to Believers, and I hope you will help us get to the next level so that we can be the best medical college in the country. You are in the best medical college. We want to get better than that. I hope you have a great, great time with us. Welcome. Thank you for joining us.
Thank you, dear friends. You may go back to your places. So, thank you all for coming to this very important session. We as a department of pharmacology and the state-of-the-art TDM facility, we expect your wholehearted support as we move towards excellence in this new field of TDM. And we expect to be #1 in the country very soon, superseding CMC Vellore. Thank you. Have a blessed day.